Hi, welcome to another video lesson with Dr. Hill in biology, and in this lesson I'm going to explain, I hope, the difference between continuous versus discrete variation in populations, and I'm going to try and do it in 10 minutes. So let's think about variation in general. Now we're going to come across variation as an important concept in our study of evolution later in our learning. But let's think about sexual reproduction. In maybe about 1 billion years ago, we had the first appearance of sexual reproduction in organisms on Earth. And what's the point of sexual reproduction? Well, the point of it is that you have an individual over here and an individual over here. And what you do is you randomly select half of the genes from this individual and randomly select half of the genes from this individual, and you put them together in a new individual. You hybridize those two parents in a new individual. And you create, what do you create? You create a completely new genome. A genome that has never existed before in the whole of history of life on our planet. And what that means is that if an organism has many offspring, every single one of those offspring are going to be different. If a population has many offspring, every single one of those offspring are going to be different. And why? Well, because we have thousands of genes. Human beings have between 30 and 40,000 genes. I don't know what the exact count is at the moment, but that's a lot of genes. And many of those genes behave in different ways depending on circumstances. So we make maybe about 1 million different proteins. So out of those genes we make 1 million proteins, and every single one of those proteins between two individuals is going to be a different set. And that's why we get a variation. And the point of it, of course, is that in a population with many genes that can all be mixed up differently, and many different versions of genes that can all be mixed up differently, we get a range of types, a range of individuals that have different traits. And depending on what the environment does, you kind of hope that some of those individuals with certain types of traits are going to be able to cope with whatever way the environment changes. If it changes one way, this bunch over here will survive. If it changes a different way, this bunch over here will survive. So this is what we call continuous variation. Continuous variation is something that a population has as a kind of a bell curve. There are a whole bunch of people in the middle, or a whole bunch of individuals in the middle that are all roughly the same. They're very close to the mean. But what you have out on the extremes is you tend to get an extreme in one direction and an extreme in the other direction. Let's take height, for example. Everybody is a different height. There may be some people that are the same height, but if we take the whole of the secondary and Westry school and line them up against the wall, and what we'll have is we will have a few very short people, and we'll have a few very tall people, and most of the rest of the students will be in the middle, or roughly the same height. This is what we call a bell curve, or a normal distribution. Now, continuous variation is really important for evolution, and it's really important to understand that. But it's very difficult to understand exactly how genes work by looking at continuous variation, because continuous variation involves the hybridization of vast quantities of genes from two parents. What we want to look at is what they call a monohybrid cross. We want to look at what happens when you just take one gene from each parent. Because that gene on that parent, the gene that you get from this parent, might be a slightly different version to the gene you get from this parent. Now, these are the same genes. For example, the gene that causes a fly's eye to be a particular color. So one parent might have that gene and it makes their eyes red, and the other parent might have the same gene and it makes their eyes white. If we take those two into that gene and we put them together so that the new individual has two copies of the gene, they might get red and white, and that might produce a pink eye, or it might produce either a red eye or a white eye. So what we're seeing here is there's only 3 possibilities in that particular case. In fact, with eye color, there's only 2 possibilities, red or white. But let's imagine there are 3 possibilities. You can have red, pink, and white. So what you get is discrete variation. You can clearly see that there are 3 types. And if I take all the offspring from those 2 flies, and maybe I'll get 30 baby flies, and I let them grow up and look at the color of their eyes, I'll get 3 groups. I'll get flies with red eyes, I'll get flies with white eyes, and maybe flies with pink eyes. So that's what we call discrete variation. It's really important to understand that this is, we're looking at this with just a single gene. And we get different versions of the gene called alleles from our parents. Now, we can extend this into studies that involve more than one gene at the same time, maybe 2 genes, sometimes 3 genes. But once you go beyond that, it becomes really complicated to study. So to follow the path of 2 or 3 genes through a population is much more difficult than just following the path of a single gene. Anyway, let's have a quick look at the summary on the board. So we'll do that. And here we have continuous versus discrete variation. Continuous variation is the result of many genes and their interaction with the environment. We get a bell curve or a normal distribution. We see many individuals in the center and a few on the edges. Usually the center is somewhere close to where the environment is now. Okay. Examples are height in humans and milk yield in dairy cows. Now, our discrete variation is the result of a single gene, maybe 1 or 2 more if we're going to look at a dihybrid cross. That means 2 genes we're going to follow the fortunes of, and trihybrid crosses, which get incredibly complicated. They're things you might do in higher biology. We get clearly grouped phenotypes. We get red eyes or white eyes, sometimes pink eyes, and examples of this are eye color in flies and blood groups in humans. So continuous variation, basically we're looking at all the genes in an individual. Discrete variation, we're just looking at 1 gene, sometimes 2. And we don't really need to go any further than that. So I hope that's helpful to you. Continuous variation is important. It's going to become important when we talk about evolution later on. Discrete variation is what we're going to be talking about next when we go and look at a monohybrid cross. So I'll see you in the next lesson, and I hope you enjoyed that, and I hope it was all done in less than 10 minutes.